Let's start with another chromatographic technique, and that is ion exchange chromatography. As per its name, there are the exchange of ions with similar charges. That means positive ions from the column are exchanged with positive ions of the mixture which we want to separate. Ion exchange chromatography, it is the example of column chromatography, because in this technique also column is prepared, and hence it is the example of column chromatography, which is based on the difference in surface charges on the molecule. There are two types of ion exchange chromatography, that is anion exchange chromatography. In this anion exchange chromatography, negative ions are exchanged between ion exchanger and the mixture. For this process, DEAE cellulose and DEAE cephadex. DEAE, it is diethyl aminoethyl cellulose, and DEAE cephadex, which are positively charged compound. These are used as a column for the separation of anionic proteins, because these positively charged columns can attract anions towards itself, while for cation exchange chromatography, in which positively charged ions are separated, here CM cellulose and CM cephadex, CM, it stands for carboxymethyl cellulose and carboxymethyl cephadex. I am sorry, here it should be negatively charged. One minute. CM cellulose and CM cephadex, these are negatively charged columns which can hold cations very easily. The principle of ion exchange chromatography is different ions like cations or anions have different capacity to undergo exchange reaction on the surface of given exchanger. Each and every ions have different capacity to undergo exchange. The capacity of an ion to undergo exchange depend upon the charge and the size of the ion in the solution. It is observed that under similar conditions, the capacity to undergo ion exchange has been found to increase with increase in charge, while it decrease with increase in the size. So we can say that if the number of charges are more and if the molecules are smaller in size, then they can get exchanged easily than the molecules which are having lower charges and the bigger size. Now, methodology, as I told you, this is again example of column chromatography. So in this also column is prepared. The column, it is a glass tube, and in this column there is an ion exchanger. Ion exchanger is filled. This ion exchanger are adsorbed or based on dextran, agarose or cross-linked cellulose, which acts as a base to adsorb the ion exchanger. Ion exchanger, it may be positively charged or it may be negatively charged based on the type of the ion which we need to separate. Now, a column is also provided with a stirrer to mix ion exchanger and a mixture of substance to be analyzed or to be separated. Now the column is, as I told you, the column is filled with an ion exchanger which may be acidic or which may be basic or positively charged or negatively charged. Now here in this example you can see ion exchanger, it is a positively charged because we want to separate the proteins with negative charge. Here first this column which is positively charged ion exchanger, it is washed with 1 molar NaCl solution. When it is washed with 1 molar NaCl solutions, all the negative chloride ions are attached here. Once it gets saturated with all the negative ions, then the protein sample with negative charge is passed through it, or the protein sample which we want to separate is passed through it. Here what happened, proteins which are negatively charged, they replaces the chloride ions and they get exchanged, the chloride ions, and they attach to the positively charged ion exchanger. Once all the positively charged and negatively charged proteins are attached, then they are washed with the elutant. Elutant, they are always negatively charged solution with high gradient or high pH. So what they do, they replaces again all the protein ions, and in this way protein samples are separated. Once the process is done, the separation is done, then the ion exchanger is finally washed with the chloride ions once again, and all these chloride ions are again attached to the positively charged ion exchanger. Here there is a regeneration. We can use this column once again because it is properly cleaned, and again we are getting this initial stage where the chloride ions are attached to the ion exchanger. So in this process, again I am repeating, to separate negative protein sample, positively charged ion exchanger are used. First it is washed with sodium chloride. So all chloride ions which are negatively charged attached to the positively charged ion exchanger. Then the protein sample which we want to separate is passed. Here what happened, protein sample which are negatively charged, they replaces the chloride ion and they attach to the ion exchanger, while positively charged protein and the proteins with no charge, they do not attach anywhere and they removed easily. Once these protein samples are attached, then again it is washed with another elutant which remove the negatively charged protein sample which can be collected in the container, and that exchanger is again washed with chloride ion to regenerate it and to reuse it. So this is the methodology of ion exchange chromatography. In this technique, as there are the exchange of ions, this is called as ion exchange chromatography. Now next chromatography, that is the affinity chromatography. As per its name, there is the affinity between the molecules which we want to separate and the column which is prepared. This technique, it has a revolutionary impact on the modern biological science. This technique is based on molecular recognition principle, and it is the example of liquid chromatography. By using this technique, or in affinity chromatography, molecules are separated by the specific ligands. In this technique, there is a use of reversible biological interaction. What do you mean by reversible biological interaction? That you will understand when we will see the methodology of affinity chromatography. Here, the separation and the analysis of specific components within a sample is done by using a ligand. Here, ligands are attached to the solid matrix by covalent bond. For example, by using affinity chromatography, we can separate enzymes with an inhibitor and antigen with an antibody. Now here you can see the techniques which is used. In the first step, to an inert porous matrix, now this is the matrix filled in the column. Again, it is the example of column chromatography. First, the column is filled with the porous inert matrix, and to this inert matrix, these ligands are attached. These are the ligands. Okay. These ligands are immobilized, or these are attached to the matrix through covalent bond. So they cannot be removed easily. Then the protein sample which we need to separate, it is passed through it. This is the protein sample with different molecules. Now the specific proteins will only bind to the ligand. Here there are different shaped proteins, triangular, square and circle. Now we can see that only triangular shaped proteins can get properly attached to this shaped ligand. The unbound protein, here these protein get bound to the ligand, while the unbound proteins are removed or washed away. These are unwanted, so these are washed away. Here, specific protein will only bind to the ligand, while other proteins will pass through the column. Non-specifically bound proteins are also removed by extensive washing of column with buffer. These are unwanted, so this can be removed by washing. Now, ligand bound specific protein, these ligand bound specific proteins are now removed or released by adding soluble ligands which compete with the immobilized ligand for specific protein, or by changing the pH of buffer or salt concentration. So in this step, once these specific proteins are attached to the ligand, then these can be removed by some other elutant or by adding some soluble ligands which compete with the ligands, and they remove the specifically bound proteins, and in this way these specifically bound proteins are collected. This technique, it can be done in a single step, and in a single step it is possible to separate specific protein from a mixture of protein. So that's why this technique is very useful. Now we'll see the applications of affinity chromatography. Affinity chromatography is used for the purification of biotin. Biotin, it is one of the cofactor of several enzymes. For this technique, avidin, it is the tetrameric protein which is deposited in the egg white of birds, reptiles and amphibians. This avidian protein has affinity for biotin. As I told you, biotin is a cofactor for several enzymes. Here, avidin, it acts as a ligand, and immobilized avidin column is used for purification of biotin containing enzymes. non-biotin molecule do not bind to immobilized avidin, while biotin only attach to the avidin, and then finally it is washed away with another soluble ligands. This technique is also used for the protein elution. Elution of protein is done by chelating agents such as EGTA or EDTA. It is also used for purification of glycoproteins, for the purification of inhibitors are used as immobilized ligand to purify the enzymes. Certain hormones are used to purify the receptors, while antibodies are used as a ligand to purify the antigen. So, this is all about affinity chromatography. Thank you so much.